Yes, sir.
Would you just upload it to the Google Classroom?
Don't do that yet.
Okay.
So I just sent out a Google Classroom assignment for in class today, but we haven't gotten to the instructions yet. So we'll get there. But thank you for reminding me to say that. Okay. I hope everybody appreciates the karyogram made out of genes. Get it? Yeah. It's pretty clever. Today we're starting to talk about DNA replication. And so like usual, a process is going to be, hopefully those of you that just came in will finish the Socrative quiz. We'll double check to see how well everybody understands the concepts that you were to have read in the textbook for today. We're going to do a little bit of practice with the information that you read for class today. All of this stems from Watson and Crick, who you've heard about before. The guys decades ago that studied DNA and discovered the structure of DNA, the double helix. And remember, throw something at me if that happens. This is one of their quotes from the paper where they published the structure of DNA, the double helix. It's written rather stiffly. It has not escaped our notice. In fact, I should probably do a British accent. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. What is it that they're meaning? What are they saying? What is it about the structure of DNA that suggests how DNA is replicated? So what's the structure of DNA? The double helix. So if I give you a structure of DNA that looks like this, with this particular nucleotide sequence, what can you tell me about the DNA strand that base pairs with this one to make the double helix? Hey, somebody's replicating DNA. TACGT. So when you have a double-stranded molecule, each strand encodes the entire nucleotide sequence of a chromosome. When you start with one molecule, if you do what to that molecule to make it from double-stranded to single-stranded DNA? If you heat it up, if you denature it, then you have two strands of DNA that you can each use to synthesize the second strand of DNA. Both strands contain the exact same genetic information. So let's see how we're doing on the quiz. So which type of replication occurs? The type of replication I just described is, let's see, semiconservative. So 70% of you remembered at least that that's the type of replication. Now, whether or not you just remember that word and know what it means might be something else. So we'll see. Okay. So semiconservative replication is what occurs. DNA polymerase replicates DNA in which polarity? On the 5' end or on the 3' end? Ooh, close. So this is one of the main misconceptions that a lot of students bring to class, and this is one of the things that we're here today to clarify. Adds new nucleotides to the 3' end. So the choice here is B. So barely more than half of you are getting this one right, but that's understandable. So if DNA polymerase adds nucleotides to the 3' end of a growing strand of DNA, which direction is it reading the template strand? I'm guessing that the answers are going to look about the same. Okay, so we'll figure that out by the end of class. So totally fine. I just wanted to show you where we are to start class. So there are a few points that are confusing. Those are the really critical points from the chapter. Have any questions or concerns at this point? So that's where we're heading today. We're going to talk about what semiconservative replication is. One of the things that I really want you to be able to do by the end of the class is to apply the principles that Meselson-Stahl used. Remember, you never have to memorize names, but this is a famous experiment, the Meselson-Stahl experiment. Use the principles they used to figure out that semiconservative replication is the type of replication that occurs to DNA. And then we'll talk about DNA polymerase, the enzyme, and how it works. We'll talk about how it replicates DNA and which direction it moves along a chromosome to do so. And it all starts with the origin of life. sperm and egg fusing, and the cell cycle. So if you have not, I'm going to do something weird since we're using tablet pedagogy. I'm going to actually ask you to raise your hands. Yeah, I know, I'm shocking. We're going to use a new technology today. It's called your arms. Who here has learned about the cell cycle in some sort of class before? Middle school, high school? Okay. So I'm not going to spend, normally I would do that on a tablet. We'd take a poll, but I figured that the result was going to come that quickly. So you don't have to belabor this point. But something that you might not have explicitly learned about the cell cycle is where our life begins in the cell cycle. So what part of the cell cycle are the sperm and the egg at when they fertilize? So up here it suggests that they're somewhere in the growth or interphase. What number of each chromosome does a sperm and an egg carry? How many copies of each chromosome? 1, right? So each of these, for example, the sperm and the egg each have a copy of, let's pick on chromosome 1 again. There's the version of chromosome 1 from the dad in the sperm, of course, and the copy of chromosome 1 that's being inherited from the mom, that's found in the egg. And then, of course, there's chromosome 2, 3, 4, 5, etc. So these gametes are haploid. They have 1 copy of every chromosome. And these are cells that are essentially in interphase, growth 1 stage of the cell cycle. Okay, so when you were born, when you were conceived, you started out in interphase. Then DNA synthesis happens. So how much DNA is present after DNA synthesis? You started having 2 haploid cells. They fuse. That makes a cell with how many copies of every chromosome? 2. So as soon as fertilization happens, you go from 2 haploid cells to a diploid cell. That's before synthesis phase, right? The growth phase, synthesis, the second growth phase, G2, and then some sort of cell division, mitosis or meiosis. So what's the ploidy after you, single-celled organism, undergo synthesis? That's the DNA synthesis part, what we're talking about today. You were diploid before synthesis. How many copies of every chromosome do you have after synthesis? You start with 2, and then they double. So what's the ploidy in G2 after synthesis phase? You started out diploid, all of your chromosomes doubled. Now you've got 4 copies of every chromosome, that's tetraploid. So there is a part of every cell cycle where your cells are essentially tetraploid. There are 4 copies of every chromosome. Did anyone ever tell you that before? It's true. Normally your cells are diploid, 2 copies of every chromosome. After synthesis, every cell is temporarily tetraploid. Every cell in you. So we've talked before about what chromosomes look like at metaphase. You know that at metaphase, that's when you can actually observe chromosomes as discrete objects, like shown up here in the bottom half of this figure. This happens to be, again, from my favorite organism, the muntjac. We can see all of the individual chromosomes here. They've been stained with different fluorescent dyes that highlight the different chromosomes. So you can see 2 copies of chromosome 1, for example, 2 copies of chromosome 2, 2 copies of chromosome 3. That's what the chromosomes look like at metaphase. We've discussed this. That's what they look like in interphase. They're just blobs. When the cell is not actively undergoing mitosis, the chromosomes decondense. They form sort of cotton ball-y domains inside the nucleus. So you're looking at an entire nucleus there circled in red. That's the whole nucleus, all the chromosomes. They've just decondensed, so they fill up the space that's the nucleus. So normally chromosomes look like these big fluffy balls of nucleic acid. It's only in metaphase that they appear as sticks stick-shaped things that we see when we look at karyograms. So with these two pieces of information, the cell cycle, what happens at fertilization, and what the chromosomes look like at two different parts of the cell cycle, during M phase, mitosis phase, they're condensed, the rest of the time they're not. Now we get to that Google assignment. So I'd like everybody to draw a graph. So first we're going to agree on the axes of the graphs. Then you guys, on your tablets, draw the graphs. What does the DNA content of a cell look like throughout the cell cycle? So if we're going to draw a graph like that, what are the axes going to be? There's going to be an x-axis and a y-axis. So what's going to be on the x-axis? Independent variable, the thing we don't control. We've got a cell going through the cell cycle. So what labels should we put on the x-axis? When does fertilization happen? Which part of the cell cycle did I say those cells are at when they fertilize? Interphase. So let's start at G1. So what's the DNA content at G1? Then we'll move into synthesis after G1. Then the second growth phase. Then some sort of cell division phase, M, mitosis or meiosis. If that's the case, what are we going to put on the y-axis? What are we measuring? What am I asking you to propose? Pardon? So what's the scale we should put on the y-axis, the amount of DNA that we're going to measure in each of these different phases of the cell cycle? What's the maximum we need to have? 4. So I said that we're at some point temporarily tetraploid. Okay, so we've got a common framework now. So in the next minute or so, well, 3 minutes, let's say, because it involves taking a screenshot and attaching to the Google assignment. Plot in what, at each of these 4 stages of the cell cycle, the DNA content of a cell would be. And then also draw a representation of what a chromosome would look like at each of those phases of the cell cycle. So maybe underneath, in the bottom here, where I've left some room, I know at metaphase, my chromosome is going to look like that. What do the chromosomes look like at all the other phases? So I'll wander and answer questions if you have any, and we'll try to come back together in about 3 or 4 minutes. So best way to start is to, plot points. Mm-hmm. Yep.
So we know there's 4 at G2, and then there's...
Right, after synthesis. So before that... Right, so if it's 4 after synthesis, then it must be how many before? So you've got twice as many there as you do before synthesis. So 2. Yep. And then 4, and then 4 and somewhere in between. Exactly. Yep, so you're on your way. Yeah. Pardon me, sorry. It's all right. If you like, that's perfect. Yeah. Right. So this is really good. Yeah. So if we're talking about the first cell cycle, then that's what it would look like. Yeah. All right. So take a screenshot of what you've got, please, and send it in.
This one should be 2, right?
Here's one of your examples. So DNA content of a cell is a function of the cell cycle. What do you think? So during growth phase, by the way, let's put a little line here. Because at least one of you said, well, are we starting at fertilization? So if we're talking about a single cell over time, then those two cells, the sperm and the egg, would be haploid. So if this blue line represents fertilization, then you would go up to diploid at the start of G1 phase. Does that make sense? You start with two haploid cells. They each have one copy of every chromosome. Start G1 with two copies of every chromosome. So we hit synthesis phase. We go up from two copies at G1, that's right, to after synthesis, we're temporarily tetraploid. We've got 4 copies of every chromosome. What's misleading about the way the chromosomes are drawn? When do we see chromosomes that look like that? At metaphase. So we're not at metaphase yet. So here, that's what an individual chromosome would look like at metaphase. What I was hoping to see at G1, for example, was there are 2 fuzzy cottony blobs of DNA that are those 2 chromosomes. They're not condensed. So it's a fine distinction, but an important one. So at G2 phase, you would actually see 4 balls of nucleic acid, the 4 copies of, in this case, for example, chromosome 1. So after synthesis, we're temporarily tetraploid. What process is this graph showing after the G2 phase? We've got 4 copies of every chromosome. Where did we wind up in this graph? How many copies of every chromosome? Let's say it's 1. I hope it's not a half. It kind of looks like a half, but we're going to assume that that's 1. So what process was it that took a cell from G2 into haploid cells again? So this is showing us meiosis. What would happen if instead we were looking at a cell that was undergoing mitosis, just simple cell division? What DNA content would that cell be at the end after M phase? So every cell division starts with how many copies of every chromosome in a regular cell? For diploid cells. So here, we started the cell cycle with 2 copies of every chromosome. Synthesis, go up to 4. After cell division, back down to 2. So if that looked like that, that would be mitosis. Mitosis is simple cell division. So you want to have 1 parent cell and 2 daughter cells that have exactly the same amount of DNA. And that's what we have. In a normal cell cycle, you start with 2 copies of every chromosome, you replicate them, you undergo cell division, and you're back at having 2 copies of every chromosome again. So life starts on the left side of this blue line with fertilization. That's special. That's having haploid cells that come together to form that first diploid cell. After that, all life is 2n to 4n to 2n to 4n, 2 copies of every chromosome. After synthesis, 4 copies of every chromosome. then back down to 2 copies of every chromosome in every cell, except for what? Which? Meiosis. So except for that really tiny fraction of your cells that are going to undergo meiosis and produce again, that is, more gametes. So that's a special case in itself, this part down there. Bottom line, almost all of our cells are always diploid. After synthesis, temporarily have 4 copies of every chromosome. This is one of the really confusing parts about looking at karyograms, and I had conversations with a lot of you before our previous exam about, well, how do you know how many copies of DNA? Are we diploid or are we tetraploid? Geneticists define the ploidy of an organism during interphase. I never learned that until after graduate school. No one ever told me that. So that's why I'm making a point of it here, is it's confusing. Because during the cell cycle, our DNA content of our cells does change. But geneticists define the DNA content during interphase. So that's where we have 2 copies of every chromosome. That's why we're called diploids. Even though our bodies contain haploid cells and tetraploid cells also. It's during interphase that our somatic cells have 2 copies of every chromosome. Yeah?
Isn't G2 part of the interface?
It is also. But what happens, so it's G1.
Okay.
Sorry, I should have been more specific. It's G1 when ploidy is defined. Thank you for clarifying that. Other questions about this? Okay, so this is the start of the concept of DNA synthesis. Our chromosomes have to replicate. Meselson and Stahl were the ones that figured out how this process works. They didn't do all of the work. There were a number of people who came up with hypotheses about chromosomes and how they would replicate. I'm just showing 2 of the hypotheses out of 3 that their experiment tested. What was the critical experimental insight that they used to perform this? In other words, why are there 2 different colors of helix up there? What did they do to the organisms they were using? They were using bacteria. Why are their chromosomes different colors? They did something special to the DNA of some of the bacteria so that they could distinguish the parental or the original chromosomes from newly synthesized chromosomes.
Was it radioactive labeling?
It was a type of labeling. In this case, they used a heavy isotope of nitrogen. So what they did was they grew E. coli in, what's the regular atomic number of nitrogen? 14. Sorry, I had to check. I knew there was a reason that there was a periodic table in here. So they grew them temporarily in heavy nitrogen 15, so 1 extra neutron. So they grow these E. coli in what we'll call heavy nitrogen. The heavy part is important in just a minute. And then after that, they move the E. coli to grow in regular nitrogen, N14. So the dark helices, the dark strands, represent those heavy N15s, and the lighter colors represent the helices that were synthesized after the E. coli were moved into a medium that only had regular nitrogen. So how does this help them distinguish the 2 hypotheses? What's the difference shown up here between those 2 hypotheses? How would you be able to tell whether it's conservative replication or semi-conservative replication that's happening? How does that differ from that? In semi-conservative replication, what's semi about it? What does semi imply, mean?
The parental helix is still there, but split up into 2 different ones.
So it's something to split into two. It's like I'm semi-sure that you're paying attention. Right? I'm kind of half sure that some of you are paying attention. So in this case, the semi and semiconservative suggest this idea, as we discussed at the beginning of class, the DNA double helix separates, and each of those two original molecules serves as a template strand for making a new second strand based on the DNA sequence of the first. Of course, you all know, you told me this in the quiz at the start of class, that this is how DNA replicates. It's not the conservative method. I'm not so concerned about your memorizing which of the methods it is that DNA uses to replicate itself. What I am interested in is, do you understand the principles behind this experiment? So if you have heavy nitrogen, what did they experimentally do to detect whether or not those molecules were present at the first round of replication, or it's these molecules? What technique, what piece of equipment in the laboratory do you think they used? It's not gel electrophoresis, although I'd like to think it was. They used a centrifuge. So you've got heavy DNA, you've got light DNA, you've got a centrifuge. So if you spin a centrifuge tube in that direction, towards the bottom that is, if the centrifugal force is pointing down in this case, where are the heavier molecules of DNA going to go? They're more dense. They're going to go farther. So you're going to have a molecule that's heavy sediment faster, so H for heavy. You'll have a light molecule, an all light, two light strands, towards the top of the centrifuge tube. So that was Meselson-Stahl's prediction. If conservative replication happens, after the first round of replication, you'll have one strand that's the original, the parent strand, here in red. Right? That strand never changes. Every generation in conservative replication, that original double helix is always preserved. There's always one original molecule. All the rest are pure copies of that original. So draw for me what you see in the first generation with semi-conservative replication. What does it look like? You've got two molecules, they're both half heavy. What are you going to draw in the centrifuge tube? Are the two molecules going to look identical, or are they going to look different? So they're both 50% heavy nitrogen, 50% light nitrogen. So what are the masses, the relative masses of these two molecules? Are they the same or different? They're the same. So they have equal mass. They're half heavy. So how many bands will we see in the centrifuge tube? Just one, representing something that's in between an all-light molecule and an all-heavy molecule. So that's the essential difference in Meselson-Stahl's experiment. If conservative replication happens, you expect to see two bands after the first generation. That would be an all-heavy band and an all-light band. But if one strand of DNA is being used as a template to synthesize the second strand, you get two half-heavy molecules, you get a single band in the centrifuge tube that's an intermediate weight. This is sort of counterintuitive, but this is the data from Meselson and Stahl's experiment. So I want you to help me interpret it and tell me, did they see what they expected? So I've labeled, you can see, three different band sizes. In this case, centrifugal force was being applied in the same orientation as on the previous slide where we were drawing cartoons. So they've labeled the three different bands, heavy, half heavy, and light. And for some weird reason, I don't know, time's moving to the left. This is how it was presented in the paper in Proceedings of the National Academy of Sciences back in 1958. Maybe time ran backwards so long ago. So after one generation, labeled up there across the top, how many generations after the E. coli were moved into light nitrogen? How many bands are there in what size? Generation 1. There's a single band. It's the half-heavy size. Right? This is what convinced Meselson-Stahl that semiconservative replication was what was happening. Because if conservative replication was happening, they would have expected, as we discussed on the previous slide, a light band and a heavy band. And that's not what they see. They see 2 double helices of DNA, or a number of them actually, but from every single cell, 2 molecules of DNA, both exactly the same molecular weight. What happened at generation 2? I don't know why they have generation 1.9. Let's not get into weird details about replication. 1.9 is about 2 generations in. What's happened since generation 1? If you compare generation 1 to generation 2, what's new in generation 2? A new band. What is it? It's an all-light band. So for the first time, we see the appearance of bands that have 2 chromosomes that were both synthesized since the experiment started. So both strands of the double helix are new. What happens after 3 generations? And after 4, and after 10, and after 20? What does this experiment eventually look like? If you drew generation infinity, there would be 1 band. Which molecular weight? Light. So after replication continues in light, normal nitrogen, eventually, how many molecules of DNA, how many strands of DNA that are heavy are left at generation infinity? And beyond, sorry. So every bacterium started with 2 strands of heavy DNA, of 1 double helix. Those 2 strands are still left at the end of time. So you get more and more light bands, which is what this looks like, right? This band that's the all light gets darker and darker and darker. And the original band of heavy goes away and eventually is replaced by half heavy. That's because those 2 original strands of DNA are still there. They're just being diluted in this pool of increasingly light DNA. Somewhere in my body, maybe, are the original 2 molecules of DNA of every one of my chromosomes that I was born with. In other words, somewhere. But all the rest of my DNA was synthesized since I was born, and that's exactly what they were seeing in this experiment. So those 2 original strands are left. We just can't see them because there's only 2 strands of DNA that were the original strands. Any questions about Meselson-Stahl? So the way I'm going to ask you to interpret this or use this information in the future is to potentially draw what would you expect if conservative replication was really how DNA replicated. What would this look like? That's one way I might ask a question about this. All right. Now to the part where there's confusion. We saw the Socrative results. The class was roughly split on which direction polymerase replicates DNA and which direction it reads the template strand. So let's imagine we start with a single double strand, a double helix of DNA. Not to be confusing, I apologize. So let's say it's 6 nucleotides. We'll just focus on those 6 nucleotides for now. There are more later. This is a really long chromosome. I'm just showing you a tiny portion of it. What's the first thing that has to happen for DNA to replicate? So it's got to denature. Helicase has to unzip the genes. So the activity of some enzymes unwinds part of the double strand. This is where we get the term replication bubble. So at an origin of replication, which is where replication starts on a chromosome, when those 2 chromosomes melt, the 2 strands melt, that forms this bubble shape. What's the nucleotide composition do you suspect is going to be present at an origin of replication? Is it going to be more, are there going to be more A's and T's or G's and C's where enzymes are trying to break apart the two strands of DNA? More A's and T's, and it's true. So one of the ways that scientists experimentally determine where origin replications are is to look for parts of the chromosomes where there are lots of A's and T's. Fewer hydrogen bonds, it's easier for enzymes to break the two strands apart. Oh, of course, and incidentally, we could fill in the bottom strand if we wanted to. Hopefully I did that correctly. So we've done this before. What have I not drawn in here yet? There's something missing. Polarity, so the primes. So if this is a normal molecule of DNA and we've drawn it in the traditional fashion, what's the top strand up there, ATA, GTC, from left to right? It's going to be 5' to 3'. So let's just add that in as notation. And that makes the bottom strand antiparallel. Okay. Now we're ready for DNA polymerase. That was something somebody else up front whispered when I asked what was the next step. So we've got polarity. We've got free hydrogen bonds hanging out here in the nucleus. What does DNA polymerase need to start synthesizing DNA? So it has to have, what is it?
An RNA primer.
It has to have an RNA primer. This is exact, where did we talk about primers before? PCR. This is PCR, except what happens in your cells, not what happens in the test tube in the laboratory. So what does DNA polymerase, the enzyme we add to PCR, need to synthesize new DNA? It needs a primer. What else does it need? It needs building blocks to make DNA. Those are the nucleotides. What else does it need? One more thing. Pardon? It needs something to copy. What's that? The template strand. Okay, so we've got a template strand drawn up here already. Presume we've got nucleotides floating around in there somewhere. So there's a special enzyme, RNA primase, that synthesizes primers. So instead of us in PCR adding primers to the solution, there's an enzyme in the cell that actually makes primers for DNA polymerase to use to replicate the DNA. So in this case, although the primers are not ever just two nucleotides, for the sake of space, I'm going to show them here. So what's opposite in A in RNA code? What base pairs with A if it's RNA? U, uracil. And opposite the G, there's a C. So there's one RNA primer. It's a short one. And then we have an A there and a G there. Okay. Critical, critical, critical. What's the polarity of those primers? So the top primer is base paired to a molecule that's 5' on the left. So what's its 3'? It's crap. I practiced this so much. So those two are antiparallel. And I screwed it up anyway. Okay, so the bottom primer from left to right is 5' to 3'. It's hydrogen bonded with the bottom strand. So all the DNA is always antiparallel. Base pair DNA, antiparallel. So there's primers that RNA primase synthesize for DNA polymerase to come grab onto and synthesize DNA. So where is polymerase at this point in time? Is it on the 5' ends or the 3' ends of the primers? It's the 3' ends. So there will be a blob of DNA polymerase, I'm just going to call it Pol, that attaches to the 3' ends of the primers. And that's the direction that DNA is synthesized. It's always addition of new nucleotides to the 3' end. The first nucleotide adds on to the 3' end of the primer. The next nucleotide adds on to the 3' end of the first nucleotide, and so forth. So the DNA in black, not in red, red's RNA. The first nucleotide that polymerase adds here on the bottom strand is a T, opposite the A. What about the top strand? What's the nucleotide that gets incorporated there? Right here. First nucleotide that polymerase enzyme is going to synthesize. What's it adding in? So A is base paired to U. That's the 3' end of the primer. So the next nucleotide is opposite the T. It's an A. So the polymerases are moving that direction, replicating the DNA. So to the left on the top strand, to the right on the bottom strand. And that's only because they add to the 3' end of the primer. So I'll leave it to you to figure out the answer to the other question from the START quiz this morning. Which direction does that mean polymerase is reading the template strand? Right, it's 3' to 5'. So this polymerase enzyme here on the bottom has grabbed onto that template strand, and it's moving to the right, which means it's moving from the 3' to the 5'. It has to be, because DNA is antiparallel. So if it's synthesizing onto the 3' end of a primer, that means it has to be moving towards the 5' end of the template strand. It's a fine point, but after class, try to convince yourself that this is true. The polymerase is reading the template strand, the black strand on the bottom, from 3' to 5' as it synthesizes from 5' to 3'. Antiparallel. So what does this mean eventually for chromosome replication? So that was just the replication of the DNA near the origin of replication. What happens as replication proceeds? Where's helicase? So we've got replication bubbles. Where are we going to find the enzyme helicase? So helicase is going to be here where it's unwinding the double strand. So there will be 2 molecules of helicase. They're also moving away from each other. They're unzipping the hydrogen bonds as they travel away from the origin of replication. That's the concept of bidirectional replication. You start replication from an origin, replication proceeds in both directions from the origin. What's the problem? Oh, yeah, I hear you about the stress. So the helicase is unwinding DNA that causes some torsional strain in the DNA that has to be resolved by yet another enzyme. If we drew this after the entire chromosome has been unzipped by helicase, right, that top strand and the bottom strand started with primers. DNA was synthesized in that direction from that primer, and in that direction from that primer. What's the big problem? How much of our chromosome has been replicated? Are these full double-stranded copies of the original chromosome? You've only got half, as I've drawn it here, of each chromosome being double-stranded. The rest is all still single-stranded. It needs to be replicated. So what's the solution to this problem? By the way, these are the leading strands. The strands of DNA that synthesize starting from the replication of origin, and replication just proceeds until polymerase literally runs off the end of the telomere. It uses that, reads from 3' to 5', the template strand, until it just zip right off the end of the chromosome, and it's done replicating. How do we replicate the other half of the chromosomes? What do we need to start this process? What do we have to do to restart DNA synthesis? What does polymerase need? It needs more primers. So in this drawing above here, although it's getting pretty busy, I'm going to add one more thing to it. As helicase is unzipping the DNA, more primers can get added. So for example, here, primase could add another primer. That would look like it's right here on the drawing below. Likewise, primase could add a primer up here, right behind the replication fork on the left, which would look like that down there. So those are the 3' ends of the primers. Those are the 5' ends. So what can polymerase do now? It grabs onto the 3' end of the primer and does what? It does replication. But what happens when it gets here? It stops because it runs into the 5' end of the previously used primer. And the same is true here. Polymerase grabs onto the 3' end, synthesizes, whack, runs into the 5' end of an already synthesized strand. So replication stops. What has to happen next? More primers, more replication, more primers, more replication. So what are these? These are the Okazaki fragments. These are the lagging strands. They're lagging because they are synthesized later in the entire process, from when time starts, from when replication starts. These are synthesized later in the process. So they lag behind the synthesis of the leading strand, which is the one that's synthesized starting from a primer at the origin until polymerase literally runs off the end of the chromosome. So that's leading strands, lagging strands. Lagging strands are also called Okazaki fragments. There's one more issue with this, but we're not going to get into that in this class anyway, which is that you never actually fully replicate, as I've drawn here, not intentionally, but I did. You never actually fully replicate the entire length of the chromosome because there's no way to synthesize a primer to replicate those first few base pairs on the lagging strand. There's no place to put a primer. There's no DNA template to synthesize a primer that's complementary to. So every generation, our chromosomes get a little bit shorter, and a little bit shorter, and a little bit shorter. Every time our cells replicate our chromosomes and our cells divide, our chromosomes get a little bit smaller. It's a big issue. So this is what we're going to talk about next class. How would you use this information you learned today about meiosis and about DNA replication to solve a problem we have in society? So most modern societies anyway, technologically advanced ones, have generally agreed it's okay to do something about genetic testing to make sure that people can have parents, hopeful parents, can have offspring that maybe don't carry certain genetic diseases, the predisposition for those diseases. So if we can do designer babies, we can use in vitro fertilization to make babies. So if we're going to do this, which we're doing now in society, how are we going to use the techniques we've already discussed and the knowledge we have from genetics to make sure, as long as we're doing it, let's do it efficiently. How are we going to use this information to test oocytes, or sperm for that matter, for genetic quality? So there's a reading assignment for next class. I didn't write it up here. It's on the syllabus. Same assignment is on the syllabus. Plus, one other thing. Please take a look at this graph and just try to interpret what the two axes represent. It should be relatively similar in certain respects to what we graphed earlier today. There's an x-axis and a y-axis. Figure out what these axes represent and come back to class and we'll start talking about this graph.